Gene to protein and introduction. And by introduction meaning we are not going to need the details of protein synthesis, but look at all the things necessary to start talking about protein synthesis. So Beadle and Tatum were the ones to figure out back in the 1940s that a certain sequence of DNA, like a segment of DNA, which we now refer to as genes, are going to code for what they thought at the time enzymes. So you can imagine if you had a biological process where you had some substance that was going to be converted in the body to a new substance. So that particular reaction is going to be dictated by an enzyme. And if that enzyme is encoded for the DNA, if you change the DNA, you might change the enzymes or knock out the enzyme's ability to do this. And so imagine if you're trying to turn the substance 1 into substance 2, you want to do that reaction, you need an enzyme to do so. If you change the genes, you end up changing the enzymes, which means you don't get this second product being produced. That's essentially what they figured out. So following in the work of Beadle and Tatum, again, they thought one gene, one segment of DNA would give you one enzyme, but we realized that it is not necessarily an enzyme that you're going to get from it, so we can further generalize it as a protein. But of course, we know that proteins can be made up of several different polypeptides, and those polypeptides can actually come from different regions of the DNA. So we are going to go with the idea that one gene gives you one particular polypeptide. Polypeptide itself might be functional or may need other polypeptides to become a functional protein. The genome size of an organism is not necessarily correlated to the number of genes, and so there can be a lot of the genome that does not actually encode for polypeptides, and therefore the size of the genome does not necessarily translate to the complexity of the organism itself. You can have very simple organisms with lots of DNA, but a lot of it not actually coding for polypeptides. The genomes have introns and exons in them. So the introns, think of them as sort of interrupting the code. They're the non-coding regions. So if you have lots of DNA, there's going to be lots of regions in there that do not code for polypeptides. The parts that do are called exons. So imagine you had your segment of DNA here. There's going to be certain parts, these introns, so these blue parts right here, that are sort of interrupting the code. And let's circle the actual coded part. So these exons here, those are the part that need to actually end up as the instructions for the polypeptide. So the DNA is going to get copied, and the exons have to be the portion that make it into the final piece that needs to go to make the protein, the instructions for making that protein. Now, just because the introns are interrupting the exons does not mean that those introns are useless. They're not junk DNA. They're often referred to as being junk DNA because they don't code for polypeptides, but that's not correct. They are not necessarily junk DNA. They do have a lot of functions as well, and we can't say that we fully understand all of their functions, but we definitely know that certain things are like little switches or promoters, different segments that help the introns get recognized, or that segment of DNA being recognized. So they definitely have regulatory functions amongst other things. So quick comparison of DNA versus RNA. We know that DNA is double helix. We know we are dealing with a deoxyribose sugar. So we've got that one OH on the bottom there instead of 2. We know that the adenine in it is going to bond with the thymine. So we have these AT pairs, and it will, for eukaryotes, stay in the nucleus. RNA, also very, very important, is going to be single-stranded. However, it can actually almost have the same functionality as a protein in terms of its ability to do stuff. It is made up of the ribose sugar, so both those 3 and 4 carbons have an OH group on them. Uracil is replaced, so the new nitrogenous base that is going to be attached to these particular nucleotides, there is no thymine. Instead, it is uracil. And it is going to be found in the nucleus. It can be made in the nucleus or assembled in the nucleus, but it is able to move outside the nucleus in order to do its job. It sort of takes the message from the nucleus. The types of RNA that we're going to be talking about, and again, feel free to look it up. It takes a while to scroll through the Wikipedia page to get to the bottom of the different types of RNA, but we are only going to be looking at 3 of them. mRNA is the one that we called messenger RNA, and it is the one that starts in the nucleus and will leave the nucleus with the actual information from the DNA. So it carries the code for the proteins from the DNA, and it is made up of what we call codons. tRNA is transfer RNA. This is the stuff that will actually, it's like a little gopher that goes and gets stuff and brings it back to where it needs to be. The things that it is supposed to be getting are the actual amino acids. Remember, we're trying to make protein here. Protein's made of amino acids. they are fetched by the tRNA. Which ones to put where is encoded by the mRNA. And so they have a anticodon that is complementary to the codons on the mRNA. So the mRNA is going to have the code, and the tRNA is going to have this anticode that matches to it. rRNA is the functional part of these three RNAs that we're going to be looking at. It's the machine of the system here. So again, we have our mRNA that carries the message. We have our tRNA that is going to be carrying these little amino acids with it. And then we have this one here, which is essentially, this is kind of what we're going to think of it as. It is the machine that puts it all together. So it's ribosomal RNA, and it works to build the actual proteins. It's the machine where the protein synthesis occurs. So this is referred to, this process of taking DNA and then getting the message from the DNA, putting it into RNA, and then turning that message into protein. This has been referred, I don't like it, but has been referred to the central dogma of the genetic code. But of course, dogma is sort of the opposite of what we do in science. And so let's not refer to it as that. Let's just call it protein synthesis. And so this involves DNA to RNA, which is called transcription. Think of it like you're scribing things, or you're reading and writing the message out. And so it's taking the message from DNA and transcribing it into RNA. And then that is translated from an informational message to a physical protein, which can then go and do things. So imagine this is the nucleus is over here on the left. And so of course you've got your DNA in the nucleus, and it has the message with it. First step to get that to be turned into protein, or use that message to make protein, is to make this, let's do it in red here, this mRNA. So it is carrying the code, and it's copied from the DNA, and it is carrying the code from the DNA. So that is the transcription part. We transcribe that message from the DNA into RNA. Then what's going to happen is the RNA is actually going to leave, and there it is there, it's left the nucleus, and it can go out into the cytosol of the cell, cytoplasm, and it is then able to be read and made, protein is able to make from its message. That is going to involve tRNA, and again, they're transferring these little amino acids. You can imagine them going out into the soup of the cell, picking up some amino acids and bringing it back. And so these little things on there are the amino acids, and they're going to take those amino acids to, let's go yellow here, this thing in the middle, which is the ribosomal RNA, rRNA, and it is the machine. It is going to read the code from the mRNA. It is going to take those tRNAs with their little amino acids, and it is going to start building this protein here. And so we end up taking the message that originally was in the DNA, going to the mRNA, transcribing it into the mRNA, taking it to this machine that works with the tRNA, reading the message from the mRNA to build a protein. So DNA is transcribed into mRNA, messenger RNA. It is written in the 3 to 5 prime direction, so we'll look at the details of that in another lesson. Complementary to the template DNA strand. So again, it is reading. So imagine this red part here is the DNA. So it has a particular code to it, and then the mRNA is going to be complementary to that strand. And so where there is a G in the DNA, there will be a C in the RNA. Where there is an A in the DNA, there will be not a T because it's RNA, there will be a U in the RNA. So DNA stays safe in the nucleus. The mRNA is able to take the message from the DNA and go out into the more risky environment of the cytoplasm, where it will then be turned, or its message will then be turned into protein. It gets read by the ribosome. So again, there's the nucleus. Less cartoonish version of the last one, but the ribosome is the machine that reads this message, uses tRNA that is actually going to grab onto these amino acids, bring them over to the machine, and they're going to start getting stuck together, and you end up with this wonderful protein. How this gets done involves understanding how DNA works. So to figure this out, there was this idea that we know that it's made up of these nucleotides, and each nucleotide we're going to represent with a letter. And so the question is, how do these letters, nucleotides, how do they actually come together to carry a message? So the idea was that based on how many types there are, because there are only 4 types of nucleotides, and they need to encode for about 20 amino acids, or 20 main amino acids. And so if there's 4 bits of information, 4 different types of information, how can you set up those nucleotides so that they will code for the amino acids? So you can imagine if it was just one nucleotide responsible for a particular amino acid, since there's only 4 nucleotides, you would only be able to label essentially 4 amino acids. If you used a 2 nucleotide, so like let's say AT meant some particular amino acid, that still would not give you enough. So if you had 3, so for example ATA, then that label for a particular amino acid would give you enough. You essentially have enough labels using the nucleotides if you use 3 of them at a time. So this is where we got our triplet hypothesis. The idea was that it probably is 3 letters at a time that encodes for each amino acid. So we need to know where the amino acids go, and it would require 3 letters of the code to tell us which amino acid we would be wanting to use, or the ribosome would be wanting to use. So this was tested by inserting different amounts of nucleotides into bacterial DNA. So imagine that this is the DNA here, and so its letters go, let's get a little bit smaller here, A, C, G, T, A, T, C, A, it's like a reading test here, T, A, T, and so on and so on. And so the idea was that, okay, maybe these 3 letters here code for this amino acid. So the amino acids are shown as little circles here. So ACG codes for one amino acid, TCA is another one, TAT is another one, CCG is another one. So the idea, the triplet hypothesis, that every 3 nucleotides gives you a particular amino acid. So they tested this out by inserting, and so you can imagine the code here, they've essentially put a G right into the code here. And so now the code is got 1 extra letter to it. And so if these ribosomes are reading the mRNA 3 letters at a time, putting this 1 extra letter in would cause what's called a frame shift mutation. All of the letters get bumped 1 to the right. And so what used to be TCA is now we inserted a G, GTC, because this former frame right here, this former 3-letter code is no longer it. It is now GTC. And what used to be TAT is now got an A there and T and an A, so now it is another nucleotide. So what they found was when they did 1 insertion, it led to an inactive protein. The protein that they originally made was completely different and it didn't work. They then tried, okay, well, what if we insert 2 letters? So they inserted, let's say, a G and an A. And so again, that bumped the rest of the code over to the right by 2 letters. And again, the protein was inactive. It did not give you an active protein. It was completely different from the original. But, and this is what proved that triplet hypothesis to be true, if you do 3 insertions, these 3 letters right here, you do get a new amino acid. But if you look at it, the next 3 letters, TCA, are the same as the original second. And TAT gets reformed, and CCG gets reformed, and so on. So all the letters have been bumped to the right, but they've been bumped by exactly 3 letters. And what that does is it repositions all of the other 3 sets of letters correctly, so that if you compare these 2 proteins, the original and the new one, and again, we've got one more here, they are pretty much the same thing. So you get an active protein. It does have 1 extra amino acid in it, but generally if it's a really long protein, that's not going to have much of an effect. And so the active protein originally is reactivated when you insert 3 base pairs to it. 1 messes everything up, 2 messes everything up, but if you do 3, it is pretty much the same protein. Therefore, they knew that this code must be getting read 3 letters at a time. So this proved that triplet hypothesis to be true. These 3 nucleotides that we're reading here, so each of these 3 that we're reading up on our code here, they are referred to as a codon. So every 3 sets of nucleotides, those 3 letters, that is 1 codon, and 1 codon gets read to make 1 amino acid. So the codons, the genetic code is written in these triplets of nucleotides, and we call these codons. Each codon codes for an amino acid. So remember, there's no spaces in this, right? This is just this long phosphate backbone of nucleotides. And we're looking at the letters representing the different nitrogenous bases that are on it. And so you imagine it is all in one go. But what our ribosome will do is it will look at this, once it's translated in mRNA, it'll look at this and it'll say, okay, I'm going to break this up into 3-letter segments to make sense of it. And then we get our proper translation into the protein. And so you can imagine this sentence, if you read it up, see the T she sets, is like how our genetic code works. 3 letters at a time. And of course, there are no spaces, but the ribosome itself reads it as 3 letters at a time, and by doing so, makes sense of the message. This led to a whole bunch of work figuring out what the actual codons are. So we knew that there was 3 of them that would give you a particular amino acid, but which 3 gave you which amino acid was figured out eventually. And so essentially this is going to give us what we call our codon table. And so you can look at any 3 letters of our messenger RNA that is going to be turned into protein. And so if there was a, I don't know, UUU part of the rest of messenger RNA, phenylalanine is the amino acid that will be translated for that UUU. And then maybe there's an AAA, and then that would be a lysine. And so we'd have a phenylalanine and then a lysine. And then maybe there's a, I don't know, GGA after that. So the code would go UUU, and then what would we say, AAA, and then it went GGA. And so your ribosome will look at those 3 letters at a time, and it'll say, okay, first we need a phenylalanine, then we need a lysine, then we need a glycine, and so on and so on. And so the ribosome will start placing the amino acids in the correct order based on the code within the mRNA strand. This is a better way to look at a codon table, much more well designed. And so you can quickly find, okay, if we go, I don't know, let's go CUU, that would give you leucine. Whereas if you had CCG, that would give you proline. And so this is a faster way of reading the codon table from the 5' end to the 3' end. Give you which amino acid would go where. These, of course, are the amino acids themselves, and again, they have their full names and then their letter short forms as well as the 3-letter short forms, but we can refer to them as their names themselves, and this is what the codons actually encode for. So finally, some characteristics of the code itself. It is continuous. There are no breaks, there's no space or anything like that. It is just a continued run of letters, and it is the ribosome that is going to read those letters 3 at a time to place the amino acids in the right spot. There is actually start codons that tell the ribosome when to start, and actually a stop codon that will end the translating of the message, and it'll stop making the protein at that. Any deletion or insertion will cause mutations, and again, as we saw, this causes a big shift in the frame, and if you add 1 or 2 amino acids, it messes everything up. But if you add 3, you only get 1 extra amino acid, which isn't that different if you've got hundreds or thousands of these amino acids. So again, imagine if this was our sentence that we were saying before, right? See the T she sets. If we were to delete this first E, then the ribosome would try to read the sentence and it wouldn't make any sense. As a ribosome can only read 3 letters at a time, so that would give it something that was completely different. It wouldn't give it a functional protein. Same thing if you added a letter, so here's an extra letter added to it. So again, what that does is it shifts the frame, the reading frame, and so when you go to read 3 letters at a time, again, it's not sensical. It'll give you something, but not what you had before. So these mutations can result into very different proteins being formed. It is redundant. There are 64 codons, but we only have 20 main amino acids, and therefore, as you saw from that chart, there can be several different codes that give you the same amino acids. And you'll notice it's always the 3rd one that is going to be the normal one that is variating, so very changing. And so you have, for example, UAA, UAG, and UGA. The first letter is the same in each one of these, and they all code for stop codons. So this gives some redundancy in how the code is written. If the A gets turned into a G, it wouldn't be such a big deal because it would also give you the same stop codon. So if you're changing the final letter there and there's some redundancy in, mutations aren't going to be quite as costly if you only had 20 codons. So it prevents drastic effects due to single mutations, and it is normally that third position that can wobble, which means it could be a different letter, whereas it would still give you the same amino acid. Leucine is a good example of this. If you do CU anything, you'll get leucine. So if you get a mutation that turns an A to a G in that particular spot, it wouldn't work in the front or the middle, but at the end there, it doesn't matter which one it is, it'll still give you leucine. So that type of mutation wouldn't be as drastic of a change. Universal. Same codons give the same amino acids regardless of species. So when you go to look at what the code is for a snail and for you, it's the same code. So you can imagine what this suggests is that if you and a snail and a tree and a bacteria are all using the exact same code, wonder why that is. And again, this looks at common ancestor idea, and it also, a little bit more practically, is that we can take the machinery from other organisms and use them for our biotech applications. We can take different machines that would normally work in bacteria, and we can apply them to work in other organisms because they're all working on the same code. They're all the same amino acids, coded for by the same letters, built with very similar machinery.